Today, this morning, we have two speakers, Joern Diedrichsen and Angela Sirigu, and we'll begin with Joern's talk.
Great, thanks.
I'm sorry, my apologies. Joern has pointed out that since his talk is quite technical, he is willing to consider questions that concern the technical aspects of the talk. Discussions, please reserve for the end, as usual.
Okay, great, thanks. I'm going to talk about how the human brain is able to learn complex motor skills. That's kind of the main interest of our lab. And particularly, I'm going to focus on the production of movement sequences, like playing the piano, for example. Now, how does the brain do that? How do we actually achieve such expertise? So what changes in motor cortical areas through the thousands and thousands of hours of practice that it takes to become an expert in this task? So there are different philosophical views in motor control that to line out how that happens. And one is that really these movement sequences are learned through rote rehearsal. So through repetition, there's like these motor engrams laid down in lower-level motor areas, like primary motor cortex or maybe subcortical areas, and these motor engrams are somehow stored and can be triggered and then automatically give rise to these highly skilled movements. Now, I want to contrast that with a slightly different view, and that's a view that is partly motivated from an evolutionary perspective. Namely that as primates we've become more and more skilled manual actors, evolution hasn't necessarily endowed us with a larger primary motor cortex to store more complex movements. Rather, what it has given us is a range of premotor areas and a range of action-related parietal areas that don't project directly to the spinal cord, but exert their influence by modulating the activity in primary motor cortex. So, in a way, I'm kind of giving the anti-Brian Troll talk here, and saying that the only interesting thing there is in motor skill learning is top-down influences on low-level circuits. So, and we want to do that by looking at functional magnetic resonance imaging data. And so what we want to do is really not just look at activity, but look at representation in these premotor areas. How do they represent these complex movements? And so we heard a couple of examples of MVPA, multivoxel pattern analysis, or representational similarity analysis. I just want to take 10 minutes to really kind of motivate why we do this type of analysis for looking at fMRI data. And I'm going to do that at a very simple example, very basic example, namely how single finger movements are represented in primary motor cortex. Okay, so this is work by Naveed Ijaz in my lab. So what we do in these experiments are kind of mind-numbingly simple. We put people in the scanner, we ask them for 8 seconds to press one finger, for 8 seconds to press another finger. We record the activity, we reconstruct the surface, the cortical surface, we project that activity onto the cortical surface, and now we can look at these activity patterns. Okay? We know exactly where we want to focus, where all the action is. That's like this knob here, this band in the central sulcus, the so-called hand knob. It's about a 2 by 3 centimeter patch of cortex. So this is here the fundus of the central sulcus that makes this characteristic band. This bulging out part of the primary motor cortex is the hand knob, and that's really where neurons sit that control hand muscles. And what I'm going to show you now are activity patterns from 3 different subjects, individual subjects, moving either the thumb, the index, their middle, the ring, and the little finger. So this is what these patterns look like. So I hope when you stare at them for a while, that you're going to realize a number of things. First of all, I hope that you realize that these patterns are highly overlapping across the fingers. So the classical pen-field view of a motor homunculus for the hand is really quite a misrepresentation of what happens in primary motor cortex. Now, the second thing that you can't see here, you need to take my word for it, is that these patterns are highly reliable. So you can give me one single pattern from a single trial and can very accurately classify which finger moved. And also, these patterns are very stable. So we can take a subject, put him half a year or a year later into the scanner, and these patterns are nearly unchanged. The next thing I hope you can appreciate from this is that these patterns look very different across the different subjects. Okay? So there's high intersubject variability. In a way, everybody has its own cortical fingerprint of finger movements in his cortex. Indeed, we can quantify this. We can estimate how much of the variance across voxels and fingers can be explained by what component. And we come up with a split up for primary motor cortex for this data set of 36% is just pure noise. This is just random trial by trial variation. The systematic variance, 1/3, is explained by a systematic group somatotopy, where you see the thumb always activating more ventral region of the primary motor cortex, and then when you go to the other fingers, activating more dorsal parts of the primary motor cortex. But it's 2/3 of that systematic variability within each subject is idiosyncratic to that single subject. So that's the part of the variance that makes all these patterns look so different across the different subjects. Okay, so when you do normal univariate analysis, you would actually take the thumb patterns and average them across subjects, the index patterns and average them across subjects. So essentially you would get rid of this informative 40% that contains a lot of information about the organization. And so representational analysis doesn't do that. Representational analysis tries to look within each subject and tries to now make some inferences about the organization that includes all both the group somatotopic organization and this idiosyncratic individual organization. So how do we do this? And the secret lies in looking at the overlap between patterns, the overlap or the similarity between these patterns. So I hope you can see that the pattern for the middle and the ring finger patterns are actually quite similar here, and that the pattern for the thumb actually looks quite distinct from those two patterns. Okay? So we can quantify this by calculating some dissimilarity measures. So we take these patterns and we take any possible pair of patterns and calculate between any possible pairs, we calculate a dissimilarity metric. For example, showing here that the thumb pattern is very distinct or very distant from the ring finger pattern, and that the middle finger pattern is pretty similar, so in this blue colors, pretty similar to the ring finger pattern. You can take different distance matrices. For example, 1 minus a correlation is a favorite. This is our current favorite here, the cross-validated Mahalanobis distance that unifies some advantages of classification analysis. So you can test statistically whether two activity patterns are actually significantly different, but it also gives a really nice and reliable quantitative measure of distance. Now, the astonishing fact, and that's really what makes me convinced that this is a valuable analysis approach in this data set, is that when you look at the representational similarity matrices now in different subjects, what you see is that they look very, very similar to each other. Indeed, if we just take these 10 distances, so the upper diagonal of each of these matrices, and correlate any random two subjects with each other, we get an average intersubject correlation of 0.9. Okay? So I can take two random people out of this room, put them in the scanner, measure the activity of the fingers. I'm nearly guaranteed that your activity maps in primary motor cortex look very, very different. But at the same time, I'm also nearly guaranteed that your representational similarity matrix are going to be nearly identical. Okay? You can quantify or visualize this organization by using multidimensional scaling and try to depict them in a spatial arrangement so that the distances are similar to the representative of the distance in pattern space. And you come up with an arrangement that looks like this, with the thumb being more distinct, the fingers orderly arranged, and the pinky, again, a little bit more similar to the thumb than the ring finger. So this is how finger movements are represented in primary motor cortex, not spatially on the cortical sheet, but in representational space. So to sum up, what I've been telling you is that the actual spatial activity patterns are very variable across subjects, but somehow magically, every subject arrives at a very similar representational structure. Now this raises two issues, I think. The first question is, well, how can that be? How do they arrive at this? And so the explanation we offer in this paper is that we fit different models to this representational structure. And the model that, a very simple model that fits the data best, is actually to record what people do in everyday life with their hands. So we record during daily activities, we look at the correlation structure between fingers, and that actually, the everyday hand use explains this type of representational structure very, very well. So there are many different solutions to how to lay out these activity patterns on the cortical sheet to arrive at this representational structure. So everybody just ends up with a different fingerprint in a way, but they all share this underlying principle. The second point that you can learn from this is a methodological one, is what level of the data do you put your analysis? Where do you make your inferences? Classical univariate analysis tries to make the inferences on this spatial pattern level, averaging activity patterns across subjects. Representational analysis looks at the activity patterns, derives the representational structure, and then make inferences on this representational level. And the hope is that that's actually a level that's more invariant and tells us more about the cortical organization than the relatively random and arbitrary spatial organization on the cortical sheet. So I want to stop here and just take technical questions if you have them. Because I'm going to use these methods now to look at sequential movements. And so if you're lost completely, then now's your chance to speak up and ask. Yes?
What would happen if you did it on a group level map? If you did your matrix, then would it look similar to what happens when you do it on the individual subjects?
Yes. That's a really good question. Yes. So if I generate a group level map and then look at the representational structure, that looks actually quite similar to the one that I get from the individual subjects. I can also do the opposite. I can try to remove the group somatotopy from each individual map and then look at the leftover ones, and that also looks like that. So both the fine-grained organization and this shared organization across subjects that must be due to some genetic gradients follows that representational principle. Yeah, great question. Yes?
So let me ask this sort of, we should understand, sort of a reductio ad absurdum question. So suppose that in fact the variability across subjects was such that in one subject this pattern is in the frontal lobe, one is in the parietal lobe, one is in the occipital lobe.
Oh, I see.
But it preserves exactly the same structure. You would get exactly the same correlations, 0.9, but you would have almost no information about brain localization.
That's right. So when I talk like this, I really talk about the macro scale on the, it's like a mesoscale on the cortex, so in the order of centimeters. So I don't want to say that it doesn't matter if it's a motor cortex or a premotor cortex, so there are clear gradients, as I will show, but within this descent area of motor cortex, the voxel by voxel organization, I think, there's a lot of inter-subject variability that we want to utilize and we want to understand. Yeah. Okay. Great. So we want to now use these representational techniques for the rest of the talk to look at movement sequences. And so we started this a few years back. This is work by Tobias Wiesler, who started this in my lab. So we teach people these short movement sequences. For example, pressing the thumb, the pinky finger, the middle finger, the index, and the ring finger. And we give them these letter strings and then let them execute these sequences as fast as they possibly can. So in the beginning, they take about 2 seconds to execute these sequences. Over 4 days, they get much better. Subjects learn anything, basically, when you pay them. So they get below 1 second. And then in the post-test, we can check at how much of this learning was really sequence-specific. So we test them on the 4 trained sequences here, and we test them on 4 untrained sequences. And you can see that a lot of the learning is general, so they're generalized to other sequences, but there's a good 150 millisecond advantage for these trained sequences. And now the question that we had, what is it in the brain of these subjects that makes them execute these sequences so much faster than these sequences? So we put them in the scanner before the post-test, and we scanned them on the 4 trained sequences, and we scanned them on the 4 untrained sequences. And so this is a paradigm. We gave them a cue, then they executed the sequence once, twice, 3 times, over the time course of 13.5 seconds, and then we just averaged the activity over this whole time window. So we really tried to get rid of all the possible temporal information that is in the fMRI signal about the sequence, and really only look at the average spatial pattern, okay? So we do that for 1 sequence and go to the next sequence in a random order, and then intersperse some rest phases to get baseline. Now, for sequences, we don't know where to look exactly, so we use a searchlight approach. This was developed, surface-based searchlight was developed together with Nick Osterhoff, who is also here. So we go on the surface, we go on a point, we draw a circle, we extract the patterns, then we derive the representational similarity matrix. We come up with some summary statistics. What I'm going to show you here, the first summary statistic is just the mean distance between all these patterns, meaning how distinguishable are these patterns from each other. And then we can map that back onto the cortex. Then we can move that searchlight 1 millimeter over, do this again, and so on, until we've built up a map of representation across the whole cortical sheet. And this is what I'm showing you here. So this is an inflated view. This is the right hemisphere, the left hemisphere, medial surface here. What you see, these are left-handed sequences. These are areas where the 4 untrained sequences that are all matched for different forces, for timing, for difficulty. So in a univariate analysis, you wouldn't see any difference between these sequences. But when you look at the fine spatial pattern, you notice that there are slight differences between the patterns, and these differences are found in contralateral motor and sensory cortex, in PMD, in dorsal premotor cortex, and in parietal areas. Okay? So these are the untrained sequences. These are the ones that they just saw the first time during the scan. When we looked at the trained sequences, what we noticed is that this encoding was much stronger, and it wasn't strong necessarily in motor areas, but in premotor areas, especially in the left SMA, supplementary motor area. I want to contrast that with the univariate approach. When you just looked at average activity, there were actually hardly any differences between the trained and the untrained sequences. Indeed, if at all, the untrained sequences gave a little bit more activity in premotor and in parietal areas here than the trained sequences. Okay? So it seems that really like learning these movement skills seem to have sharpened the tuning functions or the patterns that are associated with these different sequences in these premotor areas. Now, the key question, of course, is now we have all these different areas that seem to encode that skilled movement sequence. What are they contributing over and above what motor cortex can do? How are these sequences encoded in these areas? And those are the questions that I want to address in the rest of my talk. So I'm going to walk through 3 examples that give us kind of different glimpses about what maybe premotor areas can contribute to the skilled sequence production. And so the first one, I'm going to look at timing, so the rhythm of a sequence and the spatial sequence itself, how those are maybe represented. And so what you notice when you execute a sequence, or when you look at a skilled movement sequence, is that the behavior becomes actually pretty invariant after a while. So this is the force trace under each finger from one of those sequences. You can see the subject does it very fast within a second, but you can also see that it has this like weird pattern that the first two presses are together, then he makes a gap, then there's another press and another small gap and then another 2 presses. Okay? And this temporal structure actually emerges during training. So you see it's quite variable. This is like the inter-press intervals here. It's quite variable in the first 2 days, and then somehow crystallizes in day number 3 and 4. They kind of converge on this idiosyncratic pattern of producing the sequences. And it's quite often quite different between subjects how they actually do these sequences. Okay? So Katya Kordisheva was a postdoc in my lab, wanted to know, well, how are the rhythm that underlies this sequence production, how is that encoded in motor areas apart from the sequence? And so when you look at the literature, there are like 2 different views, contrasting views on how timing of skilled movements is represented. The one camp holds that movement timing isn't specially represented in the brain. It's inseparably encoded together with the motor program itself. So basically the question of when to press a finger and which finger to press are just encoded together and can't be separated. The other camp holds that there might be actually some independent or more abstract representation of timing in the motor system. And so this is actually work that is motivated by birdsong work, looking at nucleus HVC, which is kind of a rough equivalent of premotor cortex in the birdsong brain, where people have found very clear indications of an abstract temporal representation of the birdsong. So one characteristic of such a representation would be that once you learn a sequence or a certain timing of a sequence, certain rhythm, you should be able to take that timing and apply it to some other motor program and generalize to that. So Katya generated a task where we actually enforced a certain timing on the subjects. It's kind of like a timed response, sequence response task, where people needed to respond to letters coming up on the screen that came up in a certain arrhythmic temporal sequence. So they needed to press one finger, next finger, next finger, next finger. They're all the 5 different fingers. So they needed to predict both when to press it and which finger to press. After learning, before learning they did it reactively. After learning, they did it partly predictively, so that the force onset was already at the force onset is already before or just after the stimulus comes up. And then she looked behaviorally, where there's indication of temporal or spatial transfer. So you tested people on the trained sequences and on completely untrained sequences, and you found a nice difference in reaction time to each stimulus. Then she looked at sequences where the order of the fingers was preserved, that was trained, but the temporal structure was new, unbeknown, and she found that these actually were executed better than the totally untrained sequences. So there's some evidence that the spatial sequence is independently represented. And then she did the other option, looking at the trained rhythm, so the temporal structure is known, but the subject doesn't know which finger to press. And again, there is some advantage compared to the untrained sequences. So there seems to be behaviorally at least some indication of that this timing was actually represented. Now, we want to use fMRI and representational analysis to look at this. Could we actually decipher where these temporal and spatial representations sit? So what we did was conduct an experiment where we had 9 different conditions. Each sequence was composed of 1 or 3 spatial sequences and 1 or 3 temporal sequences. So, for example, this first row is composed of these 3 different spatial sequences, 4, 3, 5, 2, 5, 2, 1, 5, 4, and so on. You get the idea. That all show the same temporal structure. So, 1 second interval, 1.4 second interval, 1.7 second interval, and then 2 short intervals. Okay? Then the second row would take the same spatial sequences, but now pair them with a different type of rhythm, and so on. Okay? So people learned these 9 sequences over a course of 5 days, and then we put them in the scanner, and then we let them execute these 9 sequences and image their brain. Again, these 9 sequences were perfectly balanced for movement time, for difficulty, and so on. So we know no general differences between them. Now, what I'm showing you here are these little simulated activity patches just to illustrate the type of representation we're looking for. So first of all, we can just do a classical classification approach by looking at where are the areas that actually distinguish between these 9 different possible patterns. Okay? So this is just an area, where are the regions that care about which sequence, no matter which one was executed. So this is the overall encoding. Once we find areas that show that overall encoding, we can ask the question, well, how are these encoded? So for example, in an area where the spatial sequence is encoded, you would predict that the pattern for the 3 different sequences that share the same spatial sequence would be very similar to each other. And then these should be similar to each other, these should be similar to each other, and there shouldn't be any similarities for the 3 different sequences that have different spatial sequences but the same temporal sequences. Okay? The same thing for the temporal one. So here we predict that the patterns should be similar for all the sequences that had the same temporal structure. And then we can look at interaction effects, where once we subtract out the means here, there would be some nonlinear interaction. So this is really kind of the multivariate equivalent of a 2x2 ANOVA, right? So we have a main effect, we have another main effect, and we have the interaction. And like in a 2x2 ANOVA, we can now break up the overall encoding, the overall sums of squares, into these 2 main effects and the interaction. So this is really all that is. But the strength of this approach is that we can distinguish 2 very interesting types of representation. We can distinguish one where the spatial and temporal representation is integrated in a nonlinear fashion. And we can distinguish a representation where both the space and the time is encoded, but where they're encoded additively on top of each other. Okay? So we can distinguish those 2 types of representation. That will be essential when we look at the results. So this is, we did this both with the left and the right hand. So I'm showing the results with the contralateral hemisphere on the right side. So again, it's the same old friend of sequence encoding that we see here, contralateral motor and sensory cortex, premotor areas, parietal areas, and also the SMA. So that's where there's general differences between these different sequences. Another question is, how do they encode those sequences? The first thing we can look at is the integrated encoding here in red, and we show that very focally in primary motor cortex and sensory cortex. Okay? So there really, each of the 9 different sequences has its own distinct spatial pattern. Okay? So now we can go and search for spatial representation. So where is there a region that has similar spatial activity patterns for the same spatial sequences? And we find that actually the dorsal premotor cortex, also SMA, and these parietal regions clearly show evidence for such a spatial encoding, separate spatial encoding from the timing. And we can do the same thing for the timing. And we find that now overlapping areas in premotor areas, but not parietal areas. The parietal areas mostly represent the order of these sequences. Overlapping areas in premotor areas seem to also encode the timing. So this is essential. There's a huge difference between this area, where these two codes are overlaid linearly, and the motor cortex, where also time and space is encoded, but it's coded in a very different fashion. It's coded in an interactive, multiplicative fashion. Okay? We also see some differences in the time and space encoding. So ventral premotor cortex has more temporal encoding, and dorsal premotor cortex has more spatial encoding. We also find significant gradients in SMA with anterior regions more temporally and posterior regions more spatially. Okay. So to sum that part of the talk, what we find is that in primary motor cortex, there seem to be some low-level details of the movements encoded, so that each combination of these two variables, x and y, so timing and space, are encoded in a separate spatial activity pattern. Okay? And in premotor areas, we seem to have this funny decomposition that they seem to care about the temporal structure. Some voxels seem to be care about the spatial structure, but they are additively overlaid. Okay. So what might be the computational function or the usage of such a representation? So there are two answers, I think, to that. One is efficiency of encoding. So if you think about your behavioral repertoire, all the things that you can possibly do, if you try to encode all the things that you can possibly do and that you can possibly learn with an own activity pattern, you very quickly would run out of cortical space to store all these movements in, right? Because there are just too many. So that might be the function of actually decomposing them in the constituent parts. So now you can decompose these constituent parts, combine them, and have a much more efficient encoding of these complicated movements. The other thing that endows, the other issue that you get, or the other gain that you get from having such an encoding is behavioral flexibility. So now you can, for example, take a temporal code and combine that with a new melody. So like a good pianist can take a known melody and combine it with a new rhythm or vice versa and kind of start improvising and generate genuinely new motor behaviors. So for the rest of the talk, I want to focus not on the temporal representation, but on the spatial representation. So it's bringing me to the fourth experiment, which is looking at these spatial representations, asking the question, what reference frame are these spatial representations stored in? Are they stored in an extrinsic reference frame or an intrinsic reference frame? And this is kind of an old classical issue in the psychological literature. So basically, if we think about, do some linear speculation about how we play the piano, so we read some music on a sheet of paper, and then maybe the brain translates that into some spatial code. So which key to press on the piano keyboard, okay? And this would be, of course, independent of whether I execute that sequence with the left hand or the right hand. The keys and the melody are staying exactly the same. So it would be an effector-independent representation, okay? The other idea is that maybe, or the next step, that spatial code needs to be translated into some intrinsic or muscle-based reference frame. So for the left hand, you need to specify, you need to press the pinky and then the middle finger, and for the right hand, you would need to specify pressing the thumb and then the ring finger. Okay? So these are, of course, then an intrinsic reference frame, but they are highly effector dependent, so very different codes for the left and the right hand for the same sequence. So we wanted to look at this, and so we wanted to exploit the fact that we can look at behaviors of the left and the right hand, and then look at the match between the behaviors to then infer what spatial reference frame these are represented in. So we took two groups of subjects, one group that trained on the left hand, one group that trained on the right hand. We post-tested them on the trained and untrained sequences. We get a very nice sequence-specific learning effect. We also looked at transfer. So after they learned it with the left hand, they're also better for the trained sequences with the right hand than for the untrained sequences. So something during learning has happened that is laid down in an effector-independent type of representation. There is, of course, a healthy part of the learning is effector-dependent and specific to the limb that you're trained with. And so these results are pretty symmetric across the left and the right-hand trained group. So now that gives us the opportunity to now use MVPA and representational analysis to try to decipher the reference frame in which these sequences are stored. So if we are in an area that represents these sequences as in a spatial reference frame, so from left to right. And this is, by the way, also how we queued these sequences on the screen. So 1 refers to the leftmost key, and 5 refers to the most rightward key, both for the left and the right hand. And we let them execute these sequences 1, 4, 5, 3, 2, for example, both for the left and the right hand. So if we're now in a region that represents a sequence in a spatial reference frame, an extrinsic reference frame, or maybe as the numbers on the screen, we don't know that, then the activity patterns for this sequence should be similar to this activity pattern, more similar than to this activity pattern or these other unrelated activity patterns. So looking at this representational similarity matrix between the right-hand sequences and the left-hand sequences, we should find that this diagonal would be more similar than the off-diagonal. Okay? And when we search the brain for that, we find a very clear answer that PMD from rostral to caudal gradient really fully seems to show such a spatial representation of those sequences, and also some parietal areas that are also significant. Now, we also designed the experiments to look at whether we could see if they're effector-independent, intrinsic, or mirror-symmetric representations. So we designed it so that 1,4, the sequence 1,4, so pinky index finger, is the same sequence as the sequence 5,2, again, pinky index finger, just mirror reversed. So these pairs of sequences were mirror-symmetric sequences, and hardly any subject noticed that. But now we can look at when there's an intrinsic or mirror reference frame, then this activity pattern should be more similar to this activity pattern than these two unrelated activity patterns. So again, we can search the brain for these representations, and what we find is that motor and sensory areas, as you might expect, show an intrinsic reference frame, but also caudal premotor cortex and into anterior parietal cortex also shows this intrinsic reference frame. So these are areas that are in a similar activation state when I do a sequence with my left hand and the mirror symmetric sequence with my right hand. Okay? And these were more pronounced on the trained hemisphere, the hemisphere contralateral to the trained hand, than on the untrained hemisphere. Now, so we have an answer, in a way, that yes, there seems to be a spatial representation of these sequences. There seems to be mostly stored in rostral PMD, but also in caudal PMD. We have a intrinsic reference frame representation, not only in M1, but also in caudal PMD. So caudal PMD is really an area where extrinsic and intrinsic reference frames overlap and might be really related in this translation process. But also what we find is that this intrinsic reference frame isn't unique to the effector, right? There is some part of that is shared across effectors. So much of this intermanual transfer that we might see might also relate to these shared intrinsic representations. Okay, great. So the last 15 minutes I want to spend on one very, very new experiment that we did to push this a little bit further, and now not look only at spatial and temporal representation, but look at longer sequences and look to see if we can distinguish a hierarchical pattern sequence representation. So what's a hierarchical sequence representation? So motivation really comes out of classical cognitive memory research. And the question is, how is it that musicians can remember long sequences of notes and play for a whole evening without ever looking at the score? Okay? And so memory research has a very simple answer to that. Well, it's not very simple, but one answer to it, namely that they chunk those sequences. They don't remember the individual elements. They group these elements into chunks and then remember those. Okay, so there's some natural groups here of nodes that can be remembered, lessening the resources on working long-term and short-term memory. And then maybe these are also hierarchically organized, grouping these chunks into riffs, into phrases, and into whole melodies, and those can be stored much more efficiently in memory than the individual notes. So this idea of chunking clearly is motivated by memory research, but has been taken up by motor control research. And it's been argued for a long time that maybe learning chunks also can promote performance, the actual performance of these motor actions. So it's been shown, for example, that in a learned chunk, you can execute the second, third, and fourth press faster than the first press, so the chunk boundaries, and that you can also reuse chunks that you learned in one situation in a new situation. Okay, so Atsushi Yokoi, another postdoc in my lab, wanted to know, is there evidence for such an hierarchical representation in premotor areas in the human brain? And so the experiments that I've shown you so far had really short sequences, so sequences of about 5 presses. So it's pretty likely that they were encoded as a single, maybe as 2 different chunks. So we needed to do longer sequences. So he taught people 8 different sequences of 11-digit presses. So that's quite a difficult task for them to learn, so it takes a while. And we wanted to design the experiment that some of these sequences would share the same chunks or contain the same chunks, and some other sequences wouldn't contain the same chunks. Okay? Now, the problem is, of course, that we don't know how the subjects are going to perceptually chunk those or in the motor system chunk these. So we played a little bit a trick in that we enforced that chunking by the way we laid these sequences down in declarative memory in the first place. So what we did was, in the first day, we taught people different chunks. So for example, we taught them that the letter B, the chunk B, is 524. The press is 524. Okay? And they learned 8 of these different chunks. And over the course of the first day, they learned to execute these sequences faster and faster until they reached us. Okay. On the second day, we then taught them the 8 sequences. So for example, the sequence 3 with the Roman letter 3 here is being composed of the chunk E, F, G, and H. So for example, that sequence would be 3, 5, 4, 2, 1, 2, 5, 2, 1, 4, 3. Okay, so it's learning like a phone number and breaking up into area code and the next 3 and then last 4 digits. So people can do that, and you can see now as a behavioral criterion for chunking, you can look at the interpress interval. So you can look at the interpress interval preceding 3 and 4, so within chunk intervals, and then you can look at the interval between the 2 and the 5, so the between chunk interval. And what you find is that the within chunk interval are just as fast as on the first day, but then the between chunk interval, they needed to go basically back to memory, figure out, oh, what was this chunk F? Okay, 4 to 1, and then start executing. So this is this huge reaction time cost that you see that they really kind of needed to rummage around in the long-term memory to figure out what to do next. Then we abandoned the chunks and didn't mention them again in the experiment. We trained them for 3 days just to execute these sequences from memory. So they got the Roman numeral 3, and they just needed to produce these 11 presses and just got correct and incorrect feedback. Okay? And so they were actually able to reduce their overall reaction time, and most of that came from reducing these between chunk intervals. But even after the 5th day of training, there's still a very healthy separation between these different intervals. Okay? So we wanted to know, is this just a memory effect, or is there something in the motor system that has now laid down these chunks as motor elements? So in a post-test, we try to remove requirement of memory by just presenting 11 digits on the screen, okay? And just tell them, execute that sequence as quickly as you can, okay? So we let them execute the train sequences that consist of these chunks. This was, of course, not shown like this on the screen, but this is just to indicate how they learned them. And you see that there's a healthy chunk difference between the within chunk and the between chunk intervals. We also then rearranged these chunks in multiple ways. We arranged them, for example, leaving together pairs of chunks, so-called what we call super chunks. This is here A, B is together and C, D is together, but they've never executed this sequence. Right? And you can see, again, that they can reuse that knowledge of these chunks, and they show a very big difference between the within chunk and between chunk intervals. We can also just shuffle the chunks randomly. That gives this, the same result. We can also embed these known chunks into totally random sequences. So they start out, they don't recognize that there's anything that they know, but suddenly there is this 514 that they actually trained, and you can see again that actually speeds up their performance. And it speeds it up relative to random sequence where all the digits, there was no chunk that they actually learned. So you can see it's not an extra cost of going on these chunk boundaries, rather it's a benefit that the motor system can reap by utilizing known structure and just execute these faster for these within chunks. Okay? Also what you notice is that there's a difference not only between chunk and within chunk intervals, but even in the between chunk intervals, there are some between chunk transitions that they learned, for example, in these train sequences. So they know the transition between A and B, B and C, C and D. And that gives shorter reaction times than when they don't know it, for example, the transition between D and A and D and A and C, C and B. So there's not only evidence for some chunk representation in the behavior, there's also some higher level hierarchical representation that seems to be laid down that the motor system seems to utilize. So now, the challenge is how far can we push MVPA or representation analysis to see how these are, where these are represented. So that takes a little bit thinking first. So if you're in a region that just represents single-digit presses, over the course of these 11 presses, this region would drive through these 11 different activity patterns. Okay? And because we don't have any temporal resolution to resolve these, we'll just see the summed up pattern over these 11 sequences. Okay? And so we can actually predict what that would look like. And we don't even need to know these activity patterns. We can just directly predict the representational similarity matrix. And in this case, if these digit presses are all equally weighted, we would have to predict that all the sequences would be indistinguishable. So all the distances would be on average 0. And the reason for that is that the thumb is in each sequence twice, the index 3 times, the middle finger twice, and so on. Okay, so it's totally matched for the fingers. If in a region where there's a chunk representation, we would hypothesize that that region has a 1 activity state for chunk number 1, which then would provide descending input into the motor cortex, driving the motor cortex through these patterns. When this is done, it would move into another pattern, again driving the motor cortex through these 3 different presses, and so on. Okay? Again, we can't measure the individual patterns, but we can predict the similarity matrix that we would expect if a region represented it like this. And the reason for this particular representational matrix is the following. So sequence 1 and 2 contain exactly the same chunks, so chunks A, B, C, D. So they should be identical in terms of this representation. Okay? Sequence 3 and 4 have completely different chunks, E, F, G, and H, from the first 2 sequences, so they should be very distinct. And the 5, 6, 7, and 8 share 2 of the chunks with the first 2 sequences, but not the other 2, so there should be a medium level of dissimilarity. Okay? We can do the same thing for superchunk, that gives you some representation like this, and then we can do the same thing for the sequence, for the overall sequence, where we would need to predict that every sequence has its own distinct activity pattern, and they should be all equally different from each other. Okay, so those are the hierarchical representations. Now we can also have the counter-hypothesis of a non-hierarchical representation. And that has been hypothesized quite a bit, that the motor system actually just learns transition probabilities between fingers rather than having this complicated hierarchy. Okay? So these transition probabilities would be, it would learn the transition between finger 1 and 3, 3 and 5, 5 and 2, and for each of these finger transitions, it would have a different spatial pattern. Again, we can't measure these individual spatial patterns, but we can again predict the similarity matrix you would expect if a region did that, right? And the reason for that is that, for example, these 2 sequences shown in yellow, this sequence pairs, share very few of these digit transitions. And maybe sequence 1 and 2 here, which have a predicted low dissimilarity, would share a lot of these digit pairs. Okay, great. So now we can measure activity patterns in the cortical areas, and then ask the question, which one does it look most like? There's one detail that I forgot to tell you, which is the story isn't quite as simple as this, unfortunately. And the reason for that is that we assumed in this that the weighting of each of the presses is equal across the time course. And that's not true for multiple reasons. First of all, the bolt signal doesn't sum linearly, unfortunately. And it's also that there seems to be a clear neural nonlinearity. So we need to estimate those. So what we do on each hierarchical level, we need to estimate a decay factor of the first press is always the most important, and then a question of how fast do these decay away. Okay, and we can actually do that relatively reliable, giving rise to these representational similarity matrix that are predicted. So now we can look at the empirical ones and we can compare them to the predicted ones. And what we're doing in this case is not only that we find the one that fits best, but we try to express these measured dissimilarity matrix as a sum of these hypothesized dissimilarity matrix. So we can assess whether maybe premotor cortex shows a mixture of different representation, maybe a chunk representation and a sequence representation. Okay? So again, first we look at the overall encoding. These are now right-handed sequences to make it a little bit easier for the subjects. Again, we get the classical picture. Motor cortex, premotor areas bilaterally is very left hemispheric in this case. Ventral premotor cortex and parietal cortex, some SMA. So now we can go in with our representation analysis and look at how can we break these sequence representations up. And the first thing we can look at is the pure single-digit representation. And again, we find a very clear answer that it's mostly contralateral motor and sensory cortex, where that representational, well, this average distance is nearly perfectly explained by the single-digit representation. Also some evidence for these digit transitions, but no evidence whatsoever for these hierarchical schemes shown in red. Okay? So keeping that in mind, we can now look at the trunk representation. And we find evidence for that in premotor and parietal areas on the left side. So in these two areas, you can see that the representation is very well explained by this trunk representation here. Okay? Shown already. Also, these two areas show a hierarchically very high representation in that all the sequences have some unique pattern, so it might be the highest node of the representation. We didn't really find much evidence for the super trunk or this pairs of trunk representation. So again, this is now the sequence representation. It looks very similar to the trunk representation. There's a little bit more rostral maybe here and a little bit different parietal areas. So finally, we can look at the non-hierarchical representation. And this is where kind of the main surprise came in that we found a very clear hemispheric asymmetry, in that there's clear evidence for this hierarchical sequence representation on the left premotor cortex, and a very clear evidence for this non-hierarchical digit transition representation in the right premotor cortex. So you can see it here in the bar graph, trunk representation on the left, digit transition representation on the right, and you see a similar pattern, at least in the right SMA, for the digit transition. All right, good. There was a lot of information, so let me sum up. So what I've shown you is that the pattern in primary motor cortex really looks like a circuit that needs to generate the movement, that drives through the patterns that implement the low-level details of each of the movements. Okay? So then premotor areas, in the last part, I've shown you that it seems to encode different regularities in these sequences. And there seems to be this hemispheric asymmetry in that the contralateral, or maybe left, hemisphere seems to encode some hierarchical organization of it, and there's also simultaneously learned some non-hierarchical representation of these movement sequences in some other, maybe right hemispheric areas. More generally, it seems that the premotor cortex generally looks at what motor cortex does and seems to extract the more abstract level representation. And that has possibly two different advantages. One is that it can more efficiently store vast numbers of possible movements and also can flexibly recombine them into making novel movements. So now I haven't really answered the question that I set out to answer in the beginning, namely what really makes an expert. So we haven't really looked at very long training. So we've shown you some methods that might actually be used to look at this. So the question now, the critical question is now, what happens if you really train for thousands of hours? Is it that these motor skills are encoded in these low-level generating circuits that then become independent of this premotor input? Maybe this just serves as a scaffold to set up the right circuits. Or is it actually that this motor skill expertise is encoded in these more hierarchical, higher abstract representations that then endows the expert with the flexibility of combining and generating new movements, improvising pieces, and coming up with new movements pattern on the fly? So this is, I want to thank my group who did an amazing job. I mentioned some names on the way. I want to thank my grant support and you for your attention. Thanks.
Thank you. It was a beautiful talk. I've got lots of questions. I'll limit myself. So the first thing I want to ask is you've concentrated, so I'll explain. When musicians learn sequences, it's integrated with music. So there's clear musical sequence that allows them to, and there's a connection between the music and the motor pieces. That's how they remember. They don't remember the actual, so it plays in their head. And I'm wondering whether you looked at auditory and also at hippocampus, because that was, like when you think of a motor sequence that has to do with musical pieces, it plays in your head. So this is This is the main question.
Yeah, so we've done a number of things. So in training, we sometimes play sounds, not always, to the sequence. It just makes them learn faster because they have another consequence of their action that they can remember. We also studied actually professional violinists on these sequences and looked at where encoding is there, where we see encoding. We don't really see very much in auditory or temporal regions in encoding that might have very different reasons, right? It might be that these codes that are laid down there are just not as spatially organized as the ones in dorsal premotor and SMA in these premotor areas. So we haven't really found very good evidence, but I think your intuition is very good at that what you store is the consequences of your movements, and that's how you recall these sequences. And it would be nice to do experiments where you train people doing the same sequence, learning them associated with different consequences, and then see how that affects the encoding in the brain. Yeah, that would be very nice.
Wonderful talk. I feel like I have to go back to university, start over. What's going on subcortically? So your talk was very focused on the cortex, but a lot of us think there are important things going on below the surface.
Yeah, you're totally right. So that's kind of the methodological lens that you have on. So this multivariate voxel pattern analysis is very thankful to do in the neocortex. It works very well there. These tuning functions are very spatially organized in the neocortex, it seems like, in many neocortical regions. We can decode stuff from the cerebellum, we can decode stuff from the basal ganglia, but we scrape in the bottom of the barrel in terms of signal to noise there because they're much more interdigitated. They're much harder to do in these sophisticated games that we play in looking at the representational structure are much, it's really, I didn't present it because it's really quite hard to do. So we really need to go to 70, really high spatial resolution. And even then, I think it's going to be quite a stretch to do that. Yeah, but it's possible.
Thank you.
Thanks. Very nice talk. I have a question regarding the distinction between dorsal and ventral premotor cortex that you presented throughout your talk in terms of space versus time.
Yeah.
Now, I understand that your movements were simple button presses and sequences, so that's not reaching and grasping, but naturally I was reminded about the distinction between the dorsal medial and the dorsal lateral stream with a seeming preference for reaching and grasping. So I was wondering, would you argue that the distinction into time and space is the bigger underlying organizing principle that could also explain this distinction, or would you rather argue that these are two, say, organizing principles that coexist in Graziano style?
I think, yeah, I haven't really thought about that in that term. So, I mean, there are spatial representations in ventral premotor cortex very clearly. So one clear, pretty relatively simple explanation is that people in that task where they need to learn a rhythm and a sequence, they do some subvocal strategies to remember, to kind of like do this task, and they report that at least in the beginning, and maybe that's how the rhythms is learned, and it's kind of really bound to the vocal apparatus, and that's why it's more ventral. So that's a very simple explanation for this. So I'm not quite sure why reaching would be less temporal or more temporal than grasping. So I'm not sure.
Well, I'm thinking that space matters more for reaching than for grasping, whereas the timing between your digits to each other, of course, they both require space and time, but the relative importance of space and time should be different for reaching and grasping. But I mean, it's maybe a far stretch.
Yeah. Well, I think you touch on a more kind of fundamental issue in that I showed you 3 different experiments that kind of pass the representational space in 3 different ways. Right. Now, what I haven't done is really very clearly for you to overlap all of these things and ask the question, well, what the hell are the underlying principles that carve that representational space in these different things? And so that's really going to demand much more dedicated experiments to really then now look, okay, how does the spatial representation break into extrinsic and intrinsic? How does the temporal representation, and so on and so on.
Thanks.
That was great. Thank you. In searchlight analyses, there are often assumptions when one goes about the task of comparing information that's decodable of different sorts in a searchlight, there's often assumptions that are built into the analysis choices about homogeneity of the scale at which the information is present. So for example, the sequence information versus the timing information, if that is present at different scales or if there's different structure of the organization, then the ability to decode that will vary in searchlights of different size, of smoothing of different size, of voxels of different size. And so I'm wondering what you do to protect yourself from violations of those assumptions.
I can't protect myself from that. So that's, that's the limit.
But you can.
No,
you can.
I don't think you can. No, I don't think you can. So you might have noticed that I have been made no claims about that dorsal. So what the data clearly shows is that we can, the distances are bigger. We can decode better the sequences from dorsal premotor cortex than from SMA. Right? I've not made any claims about dorsal premotor cortex seem to be more specialized for sequence than SMA. I don't think you can make the claim because it is very well possible that these sequence codes are much more spatially interdigitated in the SMA than in PMD.
But you have made the claim that for a given region it cares more about one sort of information than a different sort of information.
That's absolutely right.
And you can protect yourself against violations of those assumptions. So for example, you can ask how robust are your findings in searchlights of different sizes. You can ask how robust are your findings in different kinds of smoothing, and you can ask how robust are it to collecting data at different voxel resolutions. You can do that.
No, I mean, so yes, you can. Principally, you can do that, and we do that. So we try to estimate. We're working on statistical methods and trying to estimate the spatial scale at which these type of information live. It's not a trivial issue. So, so what's out there in the literature? There are a couple of really kind of flawed statistical attempts. For example, the debate about in V1, what spatial scale do these representations live on? Are they like ocular dominance columns or they broad spatial gradients? That literature is, to my reading, that has a number of big statistical flaws in there. So they, for example, they filter. So smoothing doesn't work with these things because we have that in our distance measure. We have that inverse noise matrix in there that just undoes the filtering. If you have if you have an invertible smoothing filter in there, it's just going to give you identical results.
Right.
So that's one problem. People have been a little bit smarter than this and kind of done non-invertible filters. But even then, that non-invertible filter on activity patterns, I'm getting very technical here, but the devil is really in the detail, is that it assumes that if you're in a voxel that is out in the gray matter and a voxel that has a lot of white matter in it, they might have exactly the same tuning function, so like the same sequence or the same type of stimulus. But of course, the one that has more white matter in it will give you lower T values, the Bolt signal, than the one that sits right smack in the thankful gray matter, where it has a lot of capillaries to live on. So now if you filter the activity pattern spatially, right, then that high frequency information just in terms of laminar, where in the cortex and the depth it is totally preserved. So you cannot actually make very good inferences about this. There are statistical methods, but it's not actually trivial. But it is a big problem. I mean, so when we see less information in some area than in some others, it is always possible that they are just too interdigitated to see. And then when we see more information, that is just more spatially clustered. So absence of information is not really something that we can make inferences on. If it's present, then we can decompose it. For example, the result that in premotor cortex they are linearly overlapped, whereas it's integrated, nonlinear integrated in motor cortex. That's something that exists at that spatial scale that we can measure. I can't say anything about the spatial scales that I can't see, but at that spatial scale, that inference I think stands.
Thanks. My question has to do with the representational sort of division of labor across the hemispheres. Mm-hmm. So sometimes in your uni manual tasks, of course you see nice, precise representations in the contralateral side, but you oftentimes see similar patterns in the ipsilateral hemisphere. Yeah, so question one is, what's with that? Question two, related, is in your hierarchical analysis, you actually show that sort of the abstract representation is in the contralateral side, the most abstract.
In this one was, yes, so we only did, so as a caveat, we only did right-hand actions here. We didn't balance, we didn't
that's fine, but nevertheless, you see sort of lower order statistics represented in the contralateral hemisphere and really high order statistics in the ipsilateral hemisphere.
So I'm not quite sure. So these were right-handed sequences, and the chunk representation and superchunk representation is on the left side, so contralateral.
Okay.
And the kind of like information about digital transitions.
That's the one that I'm referring to.
Right,
the transition. Yeah, I'm interested in. Why do you have transitional information in the right hemisphere when you're performing with your right hand, and how does this transitional information become integrated in the lower order representations in the executing hemisphere?
So, okay, so there are two questions. I'll take the first question first, and I have time to think about the second one during the time. So yeah, there's not, I mean, one thing that we realized from this work is that there are tons of ipsilateral representation, very strong. So you move your individual fingers, I haven't shown this, in the ipsilateral motor cortex there's overall bolt signal suppression, but underneath that suppression there are very clear patterns that indicate which finger moved. And it's exactly the same circuits that are there for the contralateral, the mirror symmetric ipsilateral pattern. So, you know, the ipsilateral motor cortex hangs out there, has nothing to do, something happens on the other side, it just goes with a swing, and it might be, you know, it might be completely epiphenomenal, we have no idea. It's kind of hard to think that nature would put this into place and waste so much energy and presynaptic activity when it could actually shut those down, right? So maybe it has something to do. We haven't found a good, huh? It does it very thankfully in doing bimanual action. So when you move both hands, all these things are gone. And because bolt signal really reflects EPSB, so postsynaptic activity, you know, it really indicates that these circuits don't receive the same information, or at least don't have the same recurrent activity anymore. So really kind of override that stuff. So, but that's, I mean, one of the stunning things that we see in imaging, that even the ipsilateral primary motor cortex encodes a lot about the other hand, knows a lot about the other hand. Maybe that's what you would expect if you need to perform things bimanual. One hemisphere needs to know what the other one is doing at any point in time. So maybe that's, and we work on looking at MTTs and stroke, what that ipsilateral representation might do later on in recovery. So that's a question. So now the second question, that's more tricky. I haven't thought about this. This is all, 2 weeks ago that we did the analysis and saw this. So I haven't really had very much time to think about it. So first of all, I don't know if it's left-right hemisphere or contra-ipsi. So that's something that we need to, my intuition would be that it might be left-right hemispheric. It seems that according to your talk, if the left hemisphere is specialized for language, and is somehow specialized for language that is also hierarchically organized. Sentences are hierarchically organized. Maybe it kind of does that, and they have been trained in a verbal fashion in a way. These chunks had letters and so on. Maybe that led to a left hemispheric representation, and maybe then the right PMD really just picks up the other side of the coin, as it were, for this representation that is also useful. Now, it's clear that both of these, how these combine is another interesting question, but I don't see any problem in that, actually, a computational problem in that. So imagine you have a hierarchical representation here, and it predicts the next press. It's very good within chunk, right? So, drives the motor cortical area right in the right pattern within a chunk and really does quick performance. You also saw that the trained sequences had the inter-chunk intervals were faster than for when we actually break up those chunks, when we rearrange these chunks and leave them intact. And so maybe that's part of the answer comes out of right PMD, that it learned these transitions and probabilistically now also makes its guess about what the next press is and drives motor cortex in that. Motor cortex just sees what is the next digit what is the next activity pattern that I need to assume, and it doesn't care about what that region knew or how it represented these sequences when it tells what the next digit is. So that would be my answer, I guess, about the combination. I don't think it's a very strong computational problem to combine them.
This is great, Joern. As you know, we've been looking at chunking as well, but through a different lens. So we've been looking at 12-element sequence chunks or sequences over 6 weeks of training, where the investigators, we don't impose any structure. And in that case, you see spontaneous changes in the chunk structure. it's individual specific. It can occur at any time in those 6 weeks. So it's like the person's exploring this space and eventually settling in on some kind of desired chunk pattern. So I guess I want you to speculate on, if you did that kind of experiment, like you think how it would differ. Because here you're really imposing both spatial and temporal structure on the person.
Yeah, I mean, this was kind of our first foray into chunking. And so we really wanted to make sure that we had a consistent chunk structure on which we can then make representational hypotheses. Now, as I showed you in the timing, there is clearly emerging some temporal structure with gaps, even in these 5-digit sequences that, like you observe, they are idiosyncratic, everybody comes up with their own solution, and they become invariant with training, even after 4 or 5 days. So now what would I predict? I think, it's an empirical question, really. So we really need to do the experiment. I would predict that part of this very strong hierarchical organization that you see is, of course, top-down the way it was taught, and it would actually possibly the non-hierarchical organization might be much stronger and might be also present in the left hemisphere more in the case where that chunking structure really emerges spontaneously. So it's a really interesting question whether the chunks that you come up yourself that are partly caused by biomechanical optimization, whether they actually explicitly represented as chunks, or like the camp on the integrated encoding claims are just encoded in primary motor cortex in the way these patterns just evolve from one to each other. So it's just like the biomechanical necessities of switching from one figure to the other. Maybe that actually drives what this chunking structure is.
Right, so it'd be driven by not just biomechanics, but inherent synergies that are preferred by that.
So something about the feedback loop in the peripheral motor cortical feedback loop that gives rise to these structures that is just not explicitly encoded. Right?
I really enjoyed that. I was going to ask the same question that Sharon asked, in fact, so I'll have to ask a different one. So what we're seeing here is more than a snapshot, but of the result of having learned these sequences. So we see these hierarchical structures and so on. Are you able to speculate as to where these come from? What is going on when these guys are learning these sequences? What other structures in the brain, cerebella or whatever, the role of prediction that you could imagine during early learning is really important. You're getting an error correction through there. And then gradually as your error reduces, you may find now that I don't know whether it's cerebellum or not, it's no longer indicated. Do you have any sense of what kind of there's a kind of abstraction process that's going on that leads potentially to some of these hierarchical chunking structures. In some sense, as Scott said, you're projecting that onto the participant in the form of the structured learning and giving them the temporal and spatial sequences. But people also, as we know from the way in which people learn phone numbers, they will spontaneously chunk, and as Scott says, so you see people doing that. Can you speculate as to whether these same regions are implicated in that, or that happens somewhere else?
So you're asking about the early learning part, so what happens in the first few days. We've shied away from really kind of tracking learning very early on, because then performance is so variable and so different, it's very hard to control for it in the scanner and see what you see is this like performance differences. But I think I'm quite skeptical of so there is a view out there that says, okay, well, early in learning there's these pre-motor areas and then they very quickly drop out. I think that's and then it's encoded in cerebellum and basic ganglia, and it switches around in the brain. I think that's a view that is really driven by a univariate approach in looking at activity. Because if you look at activity, you see that there's a lot of activity early on, and then that kind of goes away, shifts possibly, and so on. And I think, to me, was never very intuitive why activity would shift, or why suddenly one region should totally give up responsibility for something that it has been doing and no longer does, right? And I think the MBPA data that I showed you early on argues that there's some, right by it. Come on. One more. So, so if you look at average activity, I think the conclusion would be, yes, maybe the untrained stuff relies more on parietal and premotor areas that also really activated early in learning and then goes away because it's no longer necessary. But you can see that actually these areas actually show increase in encoding of those. I mean, those neurons or these voxels still care about the sequences. I mean, they still encode them. They just do that more efficiently. So there's always the confusion about, is this region still involved in it and just does it with less bold signal, right? Or is it really dropping out and not becoming important?
But part of the reason you're seeing that is because you actually have a theory, a model, which allows you to, in a sense, separate out your spatial and temporal and do exactly the kind of pattern distance analysis you're doing, which is fantastic. I mean, I love it. Maybe if one had an equivalent kind of model as to what's going on in learning, you would see, you wouldn't do the average activity for sure. But that may just be because we lack the right kinds of model. I mean, it's clearly the case that when I learned to play some piece of music 20 years ago, and I was painfully learning the sequence by ear and sight reading, whatever. Right. Now, 20 years later, I can sit down, barely play the thing, but I can guarantee it's not the same bits of the brain that were activated back then, with that painful, slow, laborious method.
Yeah. No, I think that's the general intuition that there are these automatic regions of it. So I'm a little bit critical for accepting that as a general rule, just because that it doesn't seem very neurologically reasonable to me that there's a region that encodes these sequences early on, that it drops out. But why would a region that had nothing to do with the sequence early in learning suddenly pick this up? Who tells whom what to do when, basically, right? So that's the question. So I think understanding these learning processes, I think I'm trying to understand rather as that each region looks at what happens, what stimulus come in and what movements are produced. And at the beginning, when you don't have the right things set up, you need to kind of build a scaffold, as it was, and kind of use your voluntary circuits, the circuits that can learn any arbitrary stimulus response mapping to kind of set up these circuits. And then circuits that don't see that at all, I think they're never going to be involved. Okay, circuits that are involved in the beginning, that then are just set up to actually
just one point. I mean, in the abstraction,
I think we should stop.
Okay.
Thank you. Thank you very much for a brilliant talk. And please come back at, let's see, in an hour, promptly, so we can have the second session. Thank you. Thank you.